this on. So these two lectures we'll be looking in today is sort of the start for the vaccine part of the module. So as you've noticed, you had some intro lectures in the last 2 weeks, and now we are moving into a big module where we'll be talking mostly about vaccine development, and later on a big module where we'll talk about drug development. So at the beginning of the drug development one as well, you have a couple of basic lectures to remind you things like pharmacokinetics and dynamics and things that you look into. So that's the same we're doing here today. We're looking into the basics of immunology in the around obviously vaccine development, but I'm not expecting you to be surprised by any of this content today. You should all know this. So it's more like a refresher. So some of you might have just finished 5842. You think, oh, that's, and for some of you, maybe go back and look at some of the lectures if some of the concepts that I discussed here today are too, you can't remember very well. So as most of these lectures, the recommended readings are generally the papers that are cited, but obviously for these two lectures, it's a bit more basic information. You can also refer to Janeway. It's half of a chapter. It's quite easy to read, and then you can look at the images there to refresh some of the concepts. So for today, again, revise, the basics of vaccination. So we're going to go back to, Edward Jenner. You're all tired of him by now. But then also look into, I'll go back to make this work. Okay. Correlates of protection, which sounds like a big name, but actually you think about and we answer together, what do we look for when we give a vaccine to a patient? So what are the protection, or the markers that we're looking for protection? And these are quite important when you're looking at trials, when you're looking at clinical trials, when you look at vaccine responses. So as we progress in this unit, you'll be looking at phase 1, phase 2, phase 3 trials, and that's where those correlates of protection will come back again, and you'll be looking at data then. And then obviously, so by the end of today, basic processes of both innate and adaptive immune responses, and then correlates of protection, and also think about factors that affect protection. So it's not just how vaccines are formulated, but the response of the public as well, the vaccinee. So if we go back again, don't need to sell why vaccinations are important, but obviously the history starts even earlier than what is considered the father of immunology, Edward Jenner. It dates back more than 500 years to that, and that's when Chinese and Turks attempt to immunize people with powdering the scabs of infected patients and blowing on the nose, as you can see in this image here. So it's interesting when you look at history of vaccination that after that experience was actually seen by this woman here, Lady Mary Montagu. She traveled to the east and actually saw that and saw how beneficial it was. And she had family members who have succumbed to smallpox before, so she decided to actually vaccinate one of their kids there through this method, the variolation. Went back to England, and then in there she made public demonstrations of how that has worked, and also she vaccinated the other kid in England to show that that was safe, and she was actually putting sort of her kid to be vaccinated, or better say variolated. And Edward Jenner was just a boy at this time, so they didn't actually, he didn't learn from her, but obviously she seeded that idea in England before Jenner. So Jenner then came with the understanding, the folk traditional history or story that milkmaids, by being exposed to cowpox, which is a different virus, would then be immune to smallpox. And that's, so here is the variolation again, that was done in England and in the West. So it wasn't blown up in the nose, but it was actually recovered from scabs and then inoculated in other people. But there was a risk to that. So about 3% of the variolation, people who were variolated, they actually developed fatal smallpox. And I don't need to dwell too much on that, because you know why. You're taking a virulent strain, it's not properly measured, you're not assessing the immune status of the patient you are giving it to it. It's live, non-attenuated vaccine, so it's sort of exposure to that antigen or that pathogen, but at a lower dose. But at this time, there wasn't such a thing as infectious dose, right? Remember, if you go back to your undergrad, when we think about lethal dose and infectious dose, those 50% of the cells that are infected, given a certain amount of exposure, so ID50s. They didn't obviously do ID50s here. It was very empirical, so there was a risk to it. Anyways, with the cowpox virus, that didn't pose an issue because it would have just benign lesions sometimes. In the milkmaids, for instance, it was really only on the hands where they were touching the cows to collect the milk. So the lesion that it extracted was from Sarah Neum's hand, and then, as you know or remember, he inoculated James Philip once and then challenged him with smallpox 3 times, and he's never developed the disease. So that's again the origin of vaccination itself, because before was variolation, and the name, as you might remember, comes from the name vaccine comes from vacca, which is Latin from cow, because that's where the first vaccine came from. Over time, that virus changed, so it's not either cowpox or smallpox. It's a vaccinia virus, which is and the evolution and change to that virus is still sort of murky in history, because nobody knows when that actually happened. But the origin of the vaccinia virus has to do with horsepox as well. So it's a different virus, what we have today in the vaccine. Do we vaccinate today against smallpox? Not routinely. It's been the only actually human disease which has been eradicated. However, people that work with it, and we still have, not we as in Australia, probably US and Russia, the wars and stuff, they have tons of that stored, because it's still a risk, as in warfare and those things. So they still have that vaccine sort of stored, and there's still some research into it as well. When did you last hear about smallpox vaccination? More like last year, maybe the year before. In which context? Do you remember? It kind of protected a little bit about another disease that is around, another pox virus, Mpox. Yeah? Remember? We have Mpox still going around. So there is a correlation that people who were vaccinated with smallpox have some protection. Now, there is a vaccine for Mpox, which again is a different virus, but people who were vaccinated with smallpox show some protection as well. So we have not been, but maybe your parents and grandparents might have some protection, because again, it's part of the pox family of viruses. So they're, kind of glad they were, right? They were vaccinated, although it's not very common, Mpox, and there is some exposures, some risks that lead to more exposure to the virus. So it's not circulating itself, but we still have cases going around. So if we look at obviously it's a cornerstone of public health policy, with hygiene, and it's another thing that really changed the field of infectious diseases, in the early last century. So once the vaccinations were introduced, you can see a dramatic drop of cases in each year. So diphtheria, for instance, so the notifications here were over, say, 50,000, and then after the vaccination was introduced in 1940, you can see that decrease really rapidly. Meningococcus C, so you can see once the vaccination was introduced again, that tail. Polio, so introduction of vaccination, so it greatly reduced measles, pertussis, and Haemophilus. There are some spikes, and this has to do with various things, including social issues. So there has been a spike in other diseases like measles, for instance. This tail here is not that low. It goes up and down a little bit more often, and it depends where you're looking at. So this is data from the UK. But if you look in the US, for instance, that changes a little bit. So it's not just the vaccine, but the uptake of the vaccine is extremely important. And you discuss, and you actually have learned a little bit already when we talk about ethics, when we talk to Chris, and the perceived safety of a vaccine really influences the uptake of vaccination as well. With polio, for instance, here, so today the issue we have is the live versus inactivated polio, right? Because without the live one, it's hard to eliminate the white viruses, because that's the only way that you can have mucosal immunity with the live vaccination. But at the same time, in areas where there is a low vaccination, that virus the virus that is derived from the vaccine can become endemic and cause disease in susceptible population. So lots of countries, including Australia, we don't use the live polio vaccine anymore, but it still has its use in some places. Another thing that is part of understanding against and remembering the principles of vaccinations is herd immunity. Now you all remember this. It has been, in the beginning of the pandemic, was something that was dreamed of, right, that we would achieve herd immunity with COVID, which we know that is not possible. It's not because of the uptake of vaccine, and especially not even the vaccine, but the virus itself here play a big role with the variation of antigens. So because there are always new viruses coming in or changes to even the Omicron variant, we won't achieve that herd immunity. There are some papers that are still discussing today if it's possible, but most papers will say we can't. But what it does is that it protects individuals who cannot be vaccinated, or who do not want to be vaccinated, or those whose vaccine has waned, which they mostly waned a little bit over time. Again, it depends on the infectiousness of the virus, as we just talked about COVID, for example, and also the effectiveness of the vaccine. So the threshold to achieve herd immunity will vary from viruses to virus, from pathogen to pathogen. We know that for diseases which have an R0 or transmission that is really high, that vaccination, that threshold is much higher. So for measles, for example, we need a coverage of over 95% to actually achieve herd immunity. For other diseases, like influenza, you can have a lower threshold, 70%, 60% in some settings, and you can still protect the population. So you would depend on the pathogen and the effectiveness of the vaccine. Now here, obviously showing no vaccination, so the infection passes from individual with the disease and it spreads throughout the population. So here everybody is susceptible. Now here, when the vaccination is below the threshold, those individuals, say this one here, which is susceptible, eventually might still get, although there is only one here infected, might still get disease because the threshold has not been achieved, and that pathogen, for instance, is highly infectious. When the vaccination coverage is achieved, that individual then is protected, because it's sort of a ring, which is a term they use in epidemiology as well, that ring of protection that is caused by those high vaccination coverage. Now, obviously, there are lots of issues here, and more recent, because I think ethics on vaccinations is a lot the discussion and the involvement of the public became much higher after COVID vaccinations and mandates and those things. So with COVID, for instance, you say you need to achieve this high vaccination. You need to vaccinate your babies, let's say, because we need to achieve that. It's a very broad and tricky discussion because it involves susceptible groups that we don't have enough data yet, maybe to vaccinate. Also, the general well-being of population. So that thought can be very confrontational to think about yourself and population level. Herd immunity, we're talking about population level. So we have, this is a public measure, right? So we're thinking not as the individual. These individual here might just not want to vaccinate because they don't believe in vaccines, or vaccines are evil, or maybe they just can't be vaccinated because they're immunocompromised and this is a live vaccine, say. Right? So again, so let's go back a little bit at those basic concepts. Right? What? How did I jump all the way there to the end of the lecture? Okay, now we go. So the immunological basis, again, when we look at immunology itself, right, obviously it prevents disease by inducing an immune response. It needs to generate long-lasting and protective immunity, and it needs to provide, to induce that in the host or in the patient, neutralizing antibodies, and we know as well cell-mediated immunity. So if you remember, the role of neutralizing antibodies is to neutralize infections, so it can neutralize toxins such as the toxin vaccine, the tetanus vaccine or diphtheria. It can prevent pathogens from adhering to cells. It can opsonize pathogens to be engulfed and then not developing disease. And it can also bind other receptors in not only in bacteria but also in viruses as well, and avoid their uptake. Concepts like herd immunity and ethical consideration and public acceptance, as I mentioned before, have to be taken into consideration when we think about basis for vaccination, but this is for another discussion from another time. And obviously we know that there are lots of different pathogens that we still don't have effective vaccines, and primarily it's because most vaccines that we have at the moment, they induce very good neutralizing antibodies, but on its own, for some diseases, neutralizing antibodies are not enough. So in malaria, for example, being an intracellular pathogen, you need to have a cellular response as well. So those CD8 T cells, for instance, need to be able to recognize infected cells and kill. Schistosoma, so usually parasitic worms and intracellular pathogens such as malaria, or here TB, which is another intracellular pathogen, is bacteria. They really need a T cell response as well. And this has been one of the challenges in developing vaccines that target that cell-mediated immunity. HIV is another issue, and that's because the changes of antigen in its surface, because it changes so easily, it's highly glycosylated, the surface of the virus. So it's really, although neutralizing antibodies are effective, but you need waves and waves of new neutralizing antibodies as the virus mutate. So the approach here is not necessarily to induce a cellular immune response, but rather to maybe produce broadly neutralizing antibodies. So antibodies that will target not a particular epitope, but something that is more common amongst different types or different, so to say, variants here of the virus. So when we look, obviously it has to be safe and inexpensive, but then again, this will be discussed more in other trials. So looking at generation of immune responses and the basics of it again, a vaccine is given, vaccine antigen plus an adjuvant, which is really important to induce those danger signals, to activate those antigen presenting cells, and that will be processed and presented to both T cells in the context of MHC class 1 here, class 2 for CD4 T cells, and in the context of MHC class 1 for CD8 T cells. The activation of those cells here will lead to effector cells that can target infected cells in the future, and here would lead to differentiation, say, into Th2 and T follicular cells, which are important for further activation of B cells, and then maturation of it, isotype switching, and affinity maturation, as you remember from before. All of these cells will then will have some subclasses that will form memory cells. So both T cells will have memory cells and B cells. And here, very importantly, will be the long-lived plasma cells, which are the ones that will respond really quickly upon seeing the pathogen again and produce very effectively antibodies when it's encountered. And here is just a description of what I've said, so you can read it at home. If we look at these individual signals and cells being activated, it's kind of important you remember the basics again. So when we look at activation of APC, it's quite important. There's strong activation and for durability of vaccines. And the reason why I say that is because if you remember, if you vaccinate an individual without an adjuvant, the immune response is going to be suboptimal. So that antigen presentation, that first part, although we talk about in correlates of protection, we talk a lot about antibodies and T cell responses, but the priming of those cells here at the beginning are quite crucial. So in the next lecture, we'll talk a little bit about adjuvants, and you see how important they are in priming that immune response, in attracting, in activating those cells. Furthermore, if the APC is not properly activated, it won't express those core stimulatory signals that are necessary to activate T cells. So if you remember, MHC class 1 is presented in all of our nucleated cells, right? So they will be presenting not just pathogen antigens, but also self-antigens as well. However, the difference is that when you're presenting a self-antigen, the core stimulatory molecules are not engaged. So that dendritic cell that is presenting, I don't know, my pancreas, some of receptor, rather the self molecule to a T cell doesn't have the B7, CD40, all of those other molecules to further engage. So the T cell will engage, not activated, will go away. However, with the proper activation, so the use of those adjuvants and maturation of that dendritic cell or other APC, as a matter of fact, macrophage or B cell as well, those co-stimulatory molecules, so here for instance CD86 and CD80, you can see how it's upregulated, and that will then engage with T cell and activate those T cells. Here for instance, these adhesion molecules, so that will bring closer the cells and really make that interaction between the T and the dendritic cell, and all the homing signals as well. So these are homing signals too. So they will go to the lymph nodes and then activate those cells there. As they migrate to lymph node, the expression of that molecule in MHC class 2 in particular is more stable, and that's quite important to activate those T cells to mature B cell responses, and then obviously this is upregulated as I mentioned. So vaccines, they again must engage multiple PRRs and inflammatory pathways to activate those cells so they can activate T cells. Now, if you look at more recent studies, and this looks overwhelming, but it's just an example of how important those pathways are. So what this paper was looking at, and this is a paper from this year or late last year, and it's quite interesting how it looks into why this has appeared first. Okay, you should be able to see this graph first and then this one. But they looked at various different markers in different cells and how they responded to different vaccines. So they looked at the PBMC, so the peripheral blood cells from quite a few adults, and after the vaccination with 13 vaccines, which are in the calendar. When they looked at that, they identified 3 endotypes. So it's a low inflammatory endotype, a medium, and a high, which I apologize that this is in the front now. And what they found is that the people with a high inflammatory endotype, they have higher antibody response across all different vaccines. So the idea that that inflammation was important for further priming T cells, you can actually see that across different individuals. When you look at the different pathways here, so B cell activation, Th2 activation, immunoglobulins as well, and inflammatory pathways, you can see that it's higher in every single. There is a very good correlation of how that's higher in the higher inflammatory group, and the response is higher in that group. What was also, so they attributed that primed stage to maybe subclinical bacterial infections, because we know the bacteria, the LPS, for example, is really potent in activating that inflammatory pathways. In fact, some of our adjuvants, they are modified LPS, so it's a bit less toxic, but it has a very good potential in activating those pathways. However, what has also we have to be mindful of is that we know that older population, they have chronic infection or chronic inflammation. But that chronic inflammation, however, was not protective. So it was interesting to see that those in older people, even though there were some inflammation there, the pathways were different. So even understanding the pathway to lead to inflammation that is effective for a vaccine is important in that sense. This is from the same paper, and I did put the reference in the image when you hover over, so you can look at it. And it's just a very nice and comprehensive paper where they again looked at the different types of pathogens, look at different types of vaccines, so included not only inactivated but also live attenuated vaccine as well. And here it's just something that is, I think, is a flaw from the study, but and you learn as we go how important it is to look at the HLA side of it as well. So a lot of vaccines which are in development or have been developed, they were tested in patients, white, male, healthy, university students. So great public, that's the target that in the US, for instance, lots of students are recruited in universities. So, and the responses, we know they vary with the different HLA types, right? So some diseases, they affect more a particular race than others. So some immune responses are better in a particular race or others. So today, most studies, they are multi-centered studies, and they need to look at different ethnicities. They need to look at white, at black, at Latin America, Indian, everything, Chinese, you know, all different Asian, all different responses, because we know that HLA is extremely important how the antigen is present in that sense as well. So after some days, they just look at various different receptors, and then the hypothesis again is that the further activation of that APC will lead to a better activation of those dendritic cells or those B cells. And they looked again at other things. So here, the high inflammatory profile, where we saw a better response, maybe they were primed by those subclinical infection. So perhaps, and this again is lots of hypotheses in this field, perhaps a patient before being vaccinated could be primed, I don't know, a week earlier, 2 weeks earlier, to hype that inflammatory pathway in the responses. And these, in fact, approaches are used in animal models. And here just looking at the different variances as well, and looking at and that marker that I mentioned, that the inflammation, the chronic inflammation in adults doesn't necessarily mean that they will respond better. In fact, we know that this age or bracket is actually poor responders. When we look at T cells, again, very basic. MHC class 1 obviously is recognized by CD8. MHC class 2 recognized by CD4 T cells. And remember from last year, or from MICR5817, that you need 3 signals to activate those cells. This is very broad. Obviously, there are more signals and more co-stimulatory molecules that might be involved. And if in this lecture series, if you find something that you don't remember, just go back and look it up. But essentially, you need antigen-specific signals, so the peptide being mounted on an MHC molecule presented to TCRs, then co-stimulatory signals, which includes those activation signals, and then cytokines, which also include IL-2 here being produced by the T cells itself, and then other different cytokines which lead to differentiation. There are some signals which are important as well, such as PTL and CTLA4, which are signals that stop the activation, so for controlling and homeostasis. Now here is just looking at how they are important again in vaccine development. So in this is a very nice paper looking at the T cell responses to COVID vaccines. So again, we know there are quite a few different variants circulating, not anymore, but we know that the virus has evolved greatly from its original strain. Then some vaccines were developed looking at the spike protein, and then over time, with the changes in the virus receptor domain, the immune responses changed, because those neutralizing antibodies that were targeting a particular part of that molecule of the spike protein are no longer has no longer the same effectiveness because it has mutated. However, when you look at T cell responses, that decline is not as drastic as you see in neutralizing responses. So there is a lot of reactivity and preservation of the T cell response, even though the virus has mutated. So here, for instance, the average was 94, and then after Omicron, with all those changes, it's still quite high, 84. CD8 as well. So the T cell responses to the epitopes, they are still quite high, and they are higher than the power of those neutralizing antibodies. So this is what is believed that gives protection even after the 6 months, when we know that those antibody levels starts to wane. So it's quite interesting, and it's quite important, and there is a lot of research at the moment, and even before that actually, I'm just looking in how can we activate T cells better, because we know how important they are, not only the CD4 but the CD8 as well, targeting infected cells, and in the context of cancer as well. So in the context of vaccines against cancer, CD8 T cells are quite important. And then here again, just looking at those pathogens that I mentioned before that need that T cell response. So what vaccines mostly are trying to look at now is how to create those memory T cells. There are different memory T cells, so there's central memory, effector, and tissue resident T cells. And here in this very nice paper looks at the reactivation of tissue resident T cells, and these are important because they are here, for instance, in the skin. So they have that first contact, they have that at the forefront in mucosal sites, interacting with pathogens. So having, for instance, CD8 resident memory cells here would be quite important in targeting pathogens. So one interesting approach that was done in mouse was looking at having resident memory T cells in mucosal sites in the vagina to prevent HIV infection. So the problem with that is that if it's CD4, you might be actually facilitating infection, because that's the target, right? So they did a very interesting thing. They gave a vaccine to a mouse, and then they did a technique which is pulling those memory cells to the site they want. And they did that by putting a cream with cytokines that will attract those cells there to the vagina canal. So they injected intramuscularly in the mouse and then use the cream to attract those cells to be resident in that particular site, which is again just a mouse model, but it's quite interesting the different approaches there to try to induce that mucosal immunity, to have not only your IgAs there as well, but your effective cells and memory cells there. When we look at B cells, again, that's very basic stuff. It's how BCR or the B cell receptor, they engage with different pathogens. As you know, it can see all the molecules, not just protein, like T cells can see in the context of MHC, but B cells can see lipids, polysaccharides, and if it's a very complex peptide or very complex polysaccharide, they can actually trigger activation in some pathways as well. Which again leads to that extensive cross-linking. For long-lasting antibody responses, you need T cells. So remember, they're T-dependent and T-independent responses. So without a T cell activation, most B cells, they will produce mainly IgM, the low-affinity antibodies, and the short-lived plasma cells. So when developing a vaccine, one has to try to engage, it's quite important to engage CD4 T cells, because that interaction between a T helper and a B cell, that's what will lead to long-lived plasma cells, isotype switching, so not just the IgM, but more importantly, IgG, which lasts much longer, and IgA in mucosal sites as well. One thing that is quite important, the interaction between the two, is linked recognition. So I want you to revise this concept. And again, this is important this is one approach that has been used in 3 vaccines we have today. So the Hib vaccine, pneumococcus and meningococcus vaccine, the antigens are polysaccharides. But remember that polysaccharides on its own might not lead to the activation of those cells, those B cells, and change to the isotype switching, long-lasting living plasma cells. So the conjugation of the polysaccharides with a protein, and then it can be a diphtheria toxin or a tetanus toxin, makes that polysaccharide now has a little bit of a protein that will be sufficient to engage with the T cells, because T cells can only see proteins. So the concept of linked recognition, it's pretty much that. It's the conjugation of a protein to polysaccharides that will allow the engagement of so this B cell, for instance, has the BCR specific to the polysaccharide, but then engages via an MHC with a protein, or the toxoid protein, and receive those signals from T helper cells to survive. So the CD40, for instance, interaction between these two. And now this cell, the long-lived plasma cell, received those signals and can become a long-lived plasma cells. So this approach is called a hapten approach as well, and there are other molecules that have been studied. For instance, here beta-glucan, which is present in Candida and Aspergillus, but because it's a type of sugar, it needs to be conjugated to a protein to lead to this response. The other thing is important to be mindful is systemic versus mucosal immune responses. So live vaccines usually, especially if given orally, will lead to mucosal, to production of mucosal antibodies as well. Flu vaccine, we have one, it's licensed in the US, which is a spray in the nose that induces a lot more IgA in the lungs. So that is more protective than usually the common approaches of injecting in the muscle, in the deltoid or the gluteus or the leg. So here, for instance, looking at those two vaccines, so Salk and Sabin. So one is live, the other one is killed. And the reason again why we don't use this before is because the vaccination rate is so great that we don't have polio has been almost eradicated in most countries. So that's why the Salk or inactivated one is sufficient. But when we look at serum IgA in some areas, for example, in the gut, in the nose, it's not present here in the killed vaccine. You find in the serum, but you do not find in mucosal sites. But if you think about how do you get polio, it's orally. So the virus is going to get into the patient through the gut. So having that duodenal IgA is quite protective. And same as example as I mentioned, the flu vaccine. There are other different types of vaccines that we use. So rotaviruses, cholera, salmonella. So rotavirus is part of the calendar. These ones in Australia, they usually given to people who go travel abroad, and the nasal flu as well. So here, just looking again at the different activation levels in different locations. So intramuscular, lots of IgG, weak cellular response, poor T cell responses. Intranasal, you have mucosal immunoglobulins as well, and activation of some resident cells, some T cells. Epidermal vaccination, again IgG, but also IgA, and then activation of resident T cells as well. So the route of vaccination is extremely important when you're thinking about where and how to deliver. The vaccines we have today, orally, nasal, intradermal, subcutaneous, intramuscular, which is the most common type, and they all targeting different things. And intramuscular, for instance, it seems like it's an easy approach because one, it's the easiest one to inject. Any person can do an intramuscular injection. But if you think about, do we really have lots of dendritic cells in the muscles? We don't. So maybe subcutaneous will be more efficient. So some vaccines, they have been restudied in quotations, looking at different delivery routes that might make it more effective or not. It depends on what type of presentation you want. It depends on also the age of the patient, what type of, so there's quite a lot of range of things to be looked into. And also it dictates the need of adjuvants. So if you inject it in a site where there isn't a lot of dendritic cells, for instance, it needs to have very potent adjuvants so you attract those cells to that site. Now this we'll talk about in the next lecture, and I'm super late, but we'll talk just about different types of vaccines, very basically looking at different approaches. So you have a lecture with Alma next week about vaccine development, so I think you'll go deeper in these approaches. But there were, in the early 20th century, as you know, it was very empirical. It was attenuation, it was done in the lab. It could take like 30 years to attenuate a virus. Now there are different ways that we can use to attenuate a pathogen in a much easier and probably safer way these days, because you know which markers you're looking for virulence, and you can knock out that in the pathogen. So it's much faster to create, to delete virus genes, and then via obviously using recombinant DNA technologies. This we'll discuss in the next lecture, but looking at the different types of vaccines. So this is very fundamental information, very basic. We have live and non-live, and non-live, or called inactivation, there in quotations. They only have a component of the pathogen or the whole killed pathogen, and there are various different formulations, as you can see listed here. There are some examples that by now or by the end of the course, you'll see in the next lecture, I have the actual immunization calendar, which you won't need to remember, memorize, but you know, you will remember that MMR is a live vaccine because we talk about it so much that it should be imprinted in your head. We also know that it's very basic, so some vaccines will require booster dose. So we know that antibody levels, they decline over time. So we have a primary immune response, then an exposure to a pathogen, and then a secondary immune response is mounted. And those antibody levels, they will stay here, but then eventually they will wane again. Here showing how it depends as well on the pathogen itself. So the incubation of that pathogen in the host is important when you're thinking about that quick response, because if it's a slow pathogen, your body will have time to react, to maybe awake. Those B cells will take a couple of days, and then the patient won't even present any signs of disease at all. If a pathogen has a very short incubation time, that's a bit different, so you're looking at other type of immune responses as well. And when we think about booster doses, you always have to think about antigenic scene as well. So we've seen that in dengue vaxia. So you might have heard before, so a vaccine that is now used only in a very few countries, and you need to have been exposed to dengue before you have the vaccine, and it's not given to children anymore. So because of that there was a sort of a low level of antibody formed in the first dose. When you encounter the pathogen, that antibody, instead of neutralizing the virus, would actually facilitate the entry of that virus into cells. So that's what it has, the antigenic scene helping that. When we look at correlates of protection, so we'll discuss that as you go in looking at clinical trials as well, in phases of clinical trials. But these are some of the things that we always look at. So look for neutralizing antibodies, obviously effective functions as well. So antibody-dependent cellular cytotoxicity and also phagocytosis. So in particular for bacterial infections, level of mucosal immunoglobulins, and then the T cell responses are important in some diseases as well. So these graph here show very nicely the different sort of assays that one can do. So here, complement fixation, phagocytosis or ELISAs, even the humble HA&CHI, which is inhibition, hemagglutinin inhibition assays, are still used for flu vaccines. When we look at correlates of protection, it's not just all of those markers that we're looking and those tests that we're looking, but more recently we are looking at other things as well. So the individualities of those patients. As I've mentioned, HLA typing, for example, why that is not a very good responder, or the age of the patient. So all the markers as well, and integrating that with an omics approach. Look at metabolics as well. It's not just the prevention of infection. It can be, the aim of the vaccine might be prevention of disease. So here, for instance, BCG is one example, SARS-CoV is another example. So at first it was aimed to prevent infection, but now we know that is to prevent severe disease. So it might change because of the nature of the pathogen. When we look at vaccine efficacy, so you learn and revise more those terms later on, but we look at efficacy, which is the phase 1 and 2 trials, so how that works well in the lab under very controlled conditions. When we take out of the lab into the public, that's effectiveness, so it has lots of other variants at a population level, and then durability as well, which might be influenced by quite a lot of things. So again, complex, it depends on the antigen, pathogen, and population. So here are some of the factors that influence that. So sex, age, ethnicity, genetics, environments, including previous infections. Again, different in different immune individuals. And here are some of the things that should be looked into when you're going later on in those trials. So demographics, so the things that are prohibited to the vaccine or not very good for the vaccine in a sense, for the protection, and some things that are good. So for instance, here we know, for instance, with COVID now, we're looking at previous infections as one of the markers. So the indication for vaccinations is 6 months since you have had your last vaccine, or infection, because we all know that infection also confers protection in some sense. Immune factors, so high antibodies, quality of antibodies as well, and here are some unknowns like the T cells, which we know a bit more now, but positive, negative, and unknown effects. There are, and this is my last slide, there are some current challenges, and I want you to think about those challenges as you see in the other lectures in terms of vaccine development and different approaches. So hard to target pathogens, TB for example, HIV, because again, antigenically very variable. Quite important, these challenges. Global security, so COVID, again, but there are other diseases like Ebola, for instance, which has a very high case fatality in comparison, and in terms of outbreaks and global security vaccines, which we have one now for Ebola, is quite important. Not only these, which are sort of panic things, but also improving immune responses in aging individuals. We all aging, all populations in every country is aging, so we need to improve the immune response to those older adults as well. And then think about therapeutic vaccines, including in the context of cancer. So those CD8 T cells, for instance. Here is just looking at, when we're talking about outbreaks and threats to global security, is just looking at emerging viral infections and the impact of some of those viral infections from the from the Spanish influenza to COVID now. And you can see case fatality rates here, some very interesting comments, so have a look through. And some are extremely difficult to control because they are zoonoses as well, so it's another facet of developing a vaccine. Doesn't have a dead end. But that's so the aim again for this lecture was just to touch back on those very broad concepts and very basic immunology. And I only had, I thought, 19 slides, and I thought, easy, I can talk just half an hour, but I talked too much, so I apologize that we're a bit late. And then we take a 5-minute and go into the next lecture. So it's very useful to look into. So we can look at that together later. Okay, so on to lecture 2 today. So it's not going to be as long as the previous one. And we just, again, it's sort of basic lecture on different vaccine types and main components. So hopefully this is not new information for you. We'll revise that, main components, as well as main vaccines used in Australia. And by the end of it, it should be able to describe some vaccine types, the immunobiology and mechanism involved in each broad group of vaccines, not necessarily each vaccine itself, but you need to understand how, say, a live vaccine works versus a mRNA vaccine, so that broad understanding. And list some of the main components, including adjuvants, which for the longest time we only had aluminum derivatives, and now we can have quite a few that have been introduced in the past few years. Again, the reading, Janeway is still a good reading for that. And also I add the pink book from the CDC, and I think the best one of all of these things of the readings is still the Australian Immunization Handbook. So if you go there, there's vaccine preventable diseases. If you click in each of those vaccine preventable diseases, it takes to the vaccines that are given in Australia, including dose, what is in the vaccine. It talks about the disease itself a little bit, so it's quite a very useful resource, and it's updated, so which is also nice. Right, now I start the lecture with this huge table here, which is the National Immunization Program Schedule. And it's not here just to illustrate and to make a point, but it's also not here for you to memorize it. So it's in between the two, right? I'm not going to be asking you when is hepatitis B given. So you need to know the months that is given, on how it's given, all of those things. But here you can see that there are some vaccines which are given more than once because it needs booster doses. So those vaccines which are part of the Australian calendar, you kind of need to know that they are given here in Australia. So for instance here, hepatitis B at birth, but then it's also repeated here at 2 months, and here you can see that it's part of a vaccine which is a hexa vaccine. Hexa means 5, so it has 5 different diseases, 6, penta is 5. So 6 different diseases. Then rotavirus, live vaccine, pneumococcal and pneumococcal vaccine. So these are types of vaccines which we just talked about, conjugate vaccines. So you need to look at these and not be, ooh, I don't know what this vaccine is. Do you know what I mean? So do not memorize the calendar, but make sure you understand why these vaccines are here, and this, why, for instance, are we giving, for instance, hepatitis B at birth. There is a rationale for that. Or why do we have to give pneumococcal vaccine many times? Why do you need to prime babies, right? Here is given at 2 months, 4 months, 6 months in some children, then again 12 months, and then again 4 years, and then you see that later on in adults, in elderly patients as well. So there is a logic and a rationale for those vaccines. Also, there are different formulations, which is interesting for you to stop and have a look at. For example, when we look at pneumococcal vaccines, there are different types. So here, for example, Pneumovax is 23. So it looks at different antigens. So there's 23 antigens here. But that's different from Prevnar here, which has 13 antigens, right? This one here is a conjugated vaccine. This one here is a polysaccharide vaccine. So the way that this one goes by that theory that we just talked about before, having a toxoid antigen bound to it to promote those long-lasting B cells. Now here, see how the patient is older as well. And pneumococcal here is not linked to, it's not a conjugated vaccine. So the response that we have at 4 years of age allows to actually give that vaccine and have immune response. If you were to give much earlier, the response of for polysaccharides only wouldn't be as effective. So have that thinking when you're looking at this table as well. So take a time, read through it, read at the notes as well, because they're interesting. For instance, rotavirus, being a live vaccine here, they will excrete, rotavirus is one nasty disease that we don't have much, but the patient, when they have rotavirus, they excrete a lot of viral particles, and also vomiting. You can do projectile vomiting, and a lot of viral particles go through. Even with the live vaccine, they can excrete some viruses as well in the feces. So one thing that we tell moms when they get the kids vaccinated is just make sure you wash your hands very well. Although we are pretty much mostly immune to it, but again, that hygiene around the house, because you have live viruses coming with the feces of the baby. So a few things. So don't just keep this table, read through it. But again, you don't need to memorize that. Same with when we look at adolescent vaccination. So it's very straightforward. Look at influenza, which is pretty much recommended for all ages. Pneumococcal vaccine here again. Meningococcal, so start kissing, sharing drinks. That's when they can get meningitis again. So again, have that critical thinking while giving, meningococcal vaccine here. Is pretty much because of the kissing and the close contact they can acquire. Also later on, earlier on, HPV. So why is HPV in this part of the calendar not to your baby? And again, sex, right? Sexually transmitted virus, pretty much. So that's why we're targeting at this age here. Adult vaccination, we look again, influenza, look at pneumococcal vaccine in specific individuals, so those more susceptible to some diseases. And you see that in yellow, as you see in the previous table, indigenous population have some extra vaccines added to the calendar, and that has to do with various things, but including, for instance, conditions of living. So they live more close contact. So that their need for more protection in some senses. So booster doses are recommended to that population. Influenza, shingles here. So that's a vaccine that you only see here very later in life. So you have the equivalent vaccine much earlier, which is the varicella, right? So that's part of the MMRV vaccine, 15 months. But here you have a booster dose that's given to elderly patients, because if they have had the disease before, they might have reactivation of the virus in a dermatome and have shingles, which is quite painful. So it's recommended here later age. And then there are some special groups as well, such as pregnant women, so influenza and whooping cough, which is not just recommended to the pregnant women, but also to people in the household. So the dad as well should get, the grandparents should get. There are some additional vaccination for people with medical risk conditions. So pneumococcal is one that is often given. So in some conditions, meningitis as well, for meningococcus as well, especially people with issues with the spleen. Because remember, the spleen is really effective in removing encapsulated bacteria. So those people who have had, or they have a splenia for some malformation, or they had the spleen removed after a car accident, whatever, they are recommended to have the vaccination as well. And then influenza, age appropriate, and then some risk conditions as well. Now, as I've mentioned, so if you go to the immunization handbook, there are a few diseases that are listed there, and these are the vaccine preventable diseases list that I took straight from there, because it's quite good and very comprehensive. So if you want to use that as a resource, you just follow that address there, and you can click on different diseases and look at the, again, the vaccines and a bit of summary of the disease itself. I highlighted here vaccines that are part of the children's calendar, and the reason is because these are sort of compulsory in a sense. Nobody is forced to have, no child is forced to have a vaccine, but there are implications here in Australia with regards to, what is it called, Centrelink and other things, because the government uses that as a social initiative as well to have a very high vaccine coverage. So those vaccines, they are good to have a look at in more detail if you are interested. And some, they are part of one formulation. So when you look at measles, for instance, you will actually be looking at the MMR vaccine or the MMRV vaccine. So let's look at the different types. So you've seen this picture before. Broadly, vaccines are divided into two types, and here I added a third thing, which is passive immunization. So live vaccines, they are attenuated pathogens sufficiently to produce an immune response, but not to cause disease. You need to remember that they can replicate, and that's what makes this vaccine very powerful. They are aimed at immune-competent individuals. Obviously, people who have immune deficiencies, there are some strict contraindications sometimes because they can actually develop disease from the vaccine, and there might be a risk, which is low, of reverting to virulent strains, such as we've seen in polio. Examples of that, so MMR, MMRV, yellow fever, oral polio, BCG, Japanese encephalitis, typhoid, zoster, and rotaviruses. All of these vaccines, they are given, except from the oral polio, they are given in Australia, but some just in certain circumstances. For example, the BCG and yellow fever, you only get it if you are traveling to a country where you have endemic disease. Even the BCG itself, you only get if you are a baby and you're traveling to an endemic country. As an adult, you might not need to have it at all. But a personal example of that is my baby. When we went to Brazil last year, I vaccinated him here before we left to Brazil with BCG. So you can do that. And it protects against disseminated types of TB, not pulmonary TB as adults, but in children is quite important. Japanese encephalitis, you can get that before you travel to endemic areas here in the South Pacific, where we know we have, and now we have some cases in Australia as well. Typhoid, you can get, again, just if you're traveling to endemic countries, and the other ones, they are part of the calendar. Non-live vaccines, it's quite a broad classification because it includes everything else. So killed, whole killed, toxoid, subunits, which can be purified, recombinant or polysaccharide or peptide vaccines, so it's quite broad here. Virus particles, so the VPLs as we have in the HPV vaccine. Conjugate, as is part of subunit vaccine, so it's just another classification of a subunit. Viral vector, as we have the Ebola, or we have the SARS, the adenovirus one. Nucleic acid, so again, SARS-CoV, so mRNA, and these ones are experimental ones. Passive immunization is not really a vaccine, but I think it's important we remember that there is that. So it's a part of treatment in some cases. So some diseases you have the vaccine against that disease, but also immunoglobulin, human immunoglobulin. So that's given when the patient has not been vaccinated or there is a serious risk for that particular disease to develop. For instance, rabies, right? So rabies is not, there is one or two maybe people who have survived. So once the disease is installed, which takes time, there is pretty much no way back. So the use of immunoglobulins there to prevent the progression of the disease is quite important. There isn't a lot of research in here because in the context of infectious diseases, because to make those antibodies is quite expensive. So you have usually to pull antibodies from people who have high titers of that particular disease, and then that's a sort of anti-serum pretty much against that antigen that you're trying to do. Some examples so can be used as pre and post exposure prophylaxis, so hepatitis A and measles. So for instance, measles, you can only vaccinate 12 months. So if you have measles and there is a baby in the household, you will, they might develop the disease. So it's recommended that they are given the immunoglobulin, and also you can anticipate sometimes the vaccination, bring it forward. Hepatitis B, varicella, tetanus, diphtheria, botulism, so and RSV, so that's some examples that you can use passive immunization. So here, just again, just looking at those different vaccines again, but just with some examples. So I've already talked about these. So weakened form of the pathogen, there is a risk that can reverse to a virulent way. People who are significantly immune compromised should not receive, and here are some of the diseases that we have live vaccines. One good example is rotavirus, which we spoke already before. So that's given orally, given at these infants which are 6 months or younger, 2 or 3 doses. It depends on the brand type that is being used. So the virus will replicate in the mucosa. And it's important again that people are aware that the virus can be shed in the stool. Interestingly, that is not indicated to people with history of intussusception, which is a type of blockage of the intestine. It kind of lives on its own because of the disease itself. Rotavirus can cause that. So here's just a cute little baby receiving oral rotavirus. So Rotatec and Rotarix are the 2 different brands given in Australia, and has really decreased the number of cases. You probably haven't seen, I remember back in med school, but it's part of the Brazilian calendar now, but I remember it's so common, it's very common disease we would get in the pediatric ward was rotavirus at some certain parts of the year. So it's very common childhood disease, which is now, pretty much crossed with vaccination. Kill subunit, there's quite a few, so it varies broad. So I use hepatitis B as an example because it's the first vaccine you give at birth. So that it's important you remember. And the risk of not giving or exposing a child to hepatitis B is that there is a much higher chance of becoming a chronic disease. So it's quite a susceptible age to be exposed to hepatitis B virus. And the vaccine has changed that completely. So it contains, in general, with killed subunits, it contains an inactivated version of the pathogen, or a subunit, or a toxoid. Induces primarily humoral immunity because there isn't that replication of the pathogen in the host. Antibody levels may wane over time, and that's why when we go back to the calendar, there's quite a few boost doses recommended. But again, it's safer. So apart from the Q fever, they are pretty much all safe in immune-compromised patients. Here, looking at hepatitis B, so the recommendations is 4 doses at birth, 2, 4, and 6. There is interestingly, so this is a 2nd generation hepatitis B vaccine. Now looking at the 3rd generation is looking at putting more antigens here. But essentially is a type of VPL, or virus-like particle. So the way it's produced, so it's, the surface antigen is isolated, transfected into yeast cells, and those yeast cells produce a lot of the surface antigen of the hepatitis B virus. That way that the antigen is produced, it becomes a viral-like particle that self-assemble into this sort of, it looks like a little bit of, it resembles a little bit the structure of a virus, and it gets in the cells much easier if you only had pieces of the the antigen in a solution. That is then adsorbed to aluminium, which is the adjuvant here, and then given to the patient, and again can maybe offered as a monovalent formulation or here as the hexa one as is given here in Australia, so all together with other 5 different diseases. Talking about VPLs, I think is an interesting technology that is good you understand. So again, they're based on the virus proteins, and they assemble on its own to form a particle. They resemble the structure, size, and symmetry of viruses, and that in terms of uptake by cells, it makes it much easier. And they are non-pathogenic, so they do not carry any genetic material inside. So here again, this is a second generation HPV vaccine, and here just looking at what is up in the future, which is adding more antigens to that vaccine and making a 3rd generation. But as you can see, the protein of interest is produced usually in those expression systems by fungi. They self-assemble and then they're co-assembled into these VPLs, and they enter the host cell, the target cells, very easily. In Australia, there are 2 types. So the 9-valent one, which is the most commonly used and targets different types, including 16 and 18, which are the ones that are more related to cancer. And there is a 2-valent one that is also recommended. So intramuscular injection, we've seen in the calendar, so young adults and adolescents, people with severely immunocompromised conditions, and men who have sex with men are recommended to take this vaccine. We've seen before the logic behind polysaccharide and conjugate vaccines. So these are quite important in those very young children, because these diseases, they usually, because we are almost colonized by, we might carry that as adults, right? But we are pretty much immune because we have our immune response a lot more mature. But remember that children under 2 years old, their B cells, they don't respond very well yet to polysaccharides in general, and that's because the immune system is pretty much immature. So being vaccinated against that is quite important, because infections, although not very common, but the infections with any of these 3 pathogens can lead to very serious complications. So meningitis, sepsis, invasive disease, bacteremia, pneumonia. So it can be quite serious, the complications of these diseases. So these 3 in particular, quite important to be very early in the calendar. So here just looking at the polysaccharide activating that B cell and the antibody production. So in adults, just the extensive cross-linked by those multivalent polysaccharides is enough to activate a B cell, but in children it needs to be conjugated by a carrier protein. So those babies who are vaccinated here, remember, they also received the DTP1, the vaccine which has a toxoid, tetanus toxoid, before that. So the T cells have seen the tetanus toxoid before. So when they encounter, for instance, the HIB vaccine here, which is conjugated to a tetanus protein, there will be memory T cells that have seen that before and interact with those B cells. They now have the polysaccharides on the BCR, but have the tetanus protein in the MHC, and therefore we receive those activation signals from T cells. So conjugated to tetanus protein, and when we look at polysaccharide only, if you remember that table, it was at 4 years of age. So by then, children can mount a better response to polysaccharides, and is not necessarily conjugated. Maybe offered again in a monovalent formulation or in combination with other vaccines. Now you will have a lecture with Dr. Archer Fox, on new technologies and mRNA vaccines, so I'm not going to dwell on this. But viral vectors and nucleic acid vaccines is another type. So mRNA, so encoding proteins of interest, they are protected by a nanoparticle which surrounds the mRNA, and that's because we have a lot of RNases. So it degrades quite fast, so it needs to be protected. They enter cells, and those cells then will start to produce, to encode for that protein, and then express in MHC class I molecules, and then will attract antigen-presenting cells, etc. Those lipidic nanoparticles, they're carriers, they are adjuvants as well, so they will attract antigen-presenting cells to the site. Recombinant adenovirus vaccine, so that is gene expressed in an adenovirus from a primate that's non-human primate, for example, chimpanzee and gorillas, and that's because if it was in an adenovirus that causes infections to humans, you might have immunity against it already because you might have been exposed before. But very rarely people are exposed to chimpanzee or gorilla adenoviruses, so therefore you don't have immunity to that yet. And examples of that is Covishield, so the Oxford AstraZeneca vaccines, which are not being used. And so we're not going to discuss the pros and cons of each of those vaccines, but I think it's important to highlight how COVID vaccines have evolved in the context of vaccine development, right? So there are a lot more COVID vaccines. So if you look at the approaches, there are quite a few that have been attempted, from inactivated vaccine to live attenuated, viral vaccines, protein subunit, which is what we use here in Australia now is this one, and the mRNA, DNA vaccines, and VPL, which is a type of protein subunit. So more different routes have been trialed, which again, from the perspective of vaccine development, is quite interesting. So intramuscular, inhalation, electroporation, and oral route. So again, open a plethora of different approaches for future vaccines to be developed as well. Different strategies, so booster with a homologous vaccine, booster with a heterologous vaccine. So again, you receive maybe 2 Pfizers, 3 Pfizers, or maybe you receive an AstraZeneca first and then you receive a Pfizer one. So that's what it has to do with heterologous and homologous. Maybe you use a protein one, then an mRNA one, and see how those combinations. So there are quite a few studies looking at those different combinations. Vaccination with people with previous infection, looking at how that induces higher immunity. So different situations and so many studies that is really hard to actually sift through these days what is good or not, but multi-centered studies as well and various different approaches. More vaccine development, so looking at polyvalent vaccines, which is what is recommended at the moment, and then again various different optimization of current vaccines to make it longer lasting or more effective, or to induce more immune response in terms of priming those early responses. So all of that, when we look into perspective, just at the vaccine development point of view, is quite interesting. Now going back to the different types of vaccines, again, just touching on this so you remember, it's not necessarily a vaccine, but we do have for some diseases a post-exposure and pre-exposure prophylaxis. So these will not induce memory. Remember, we are not activating B cells here, nor T cells. You're giving the immunoglobulin to neutralize whatever is there, the pathogen, the toxin. So it's a drug, it's a medicine, it's just the immunoglobulin you're giving, neutralizes, it resolves the patient, and then that won't lead to memory. And if that patient is exposed to that again without being vaccinated, you will need an immunoglobulin again. Here is just an illustration what would happen for rabies for terrestrial animal exposure, like a rabid dog, for example. Remember that you can get rabies from flying foxes, from bats as well, so there is a different protocol to that. But what is interesting is that there are different categories of it. And that will lead to clear, so if no prophylaxis is needed or if it's not indicated, but in some cases you will need to give the human immunoglobulin to patients here. And also you will give the vaccine to the patient. So both are given here. The immunoglobulin is not indicated only if it's been 7 days past the vaccine, because then you interfere obviously in the process of developing that immunity. But in some cases you will start obviously here, because for rabies, for instance, it's a very serious disease, so you need to neutralize that very quickly. You co-administer with a vaccine as well, and then you follow up with further vaccination. But it's just, I just find a lot of protocols for drugs and treatments, so that is just an interesting one if you want to read through. Okay, very quickly on adjuvant. The name itself, it says, it boosts, it enhances the immunogenicity of vaccines when administered. So here, vaccine without adjuvant, you have an immune response that is lessened as compared to vaccines with adjuvant. So there is a lot more APCs being coming to the site of injection, a lot more activation in draining lymph nodes, and a lot more activation of antibodies, memory B cells, and cytotoxic T cells as well. They are divided broadly into two different types, immunostimulants, so that provides those danger signals that will attract the cells, and delivery systems, which can be both, can be also immunostimulant, but also usually protects the vaccine particle to be delivered safely to the particular cell target. And one example of that is, for instance, how the lipid nanoparticle protects the mRNA in COVID vaccines. Here is just looking at how those are delivered to cells. So they're different. They can stimulate pumps, so pathogen-associated. They can be pumps that will trigger those PRRs, dumps, and small molecule agonists, or delivery systems, for instance, the lipid nanoparticles and some other formulations of it. They will enter cells in different ways. You don't need to memorize that. But again, you would see that eventually they will lead to the activation of that cell and expression of co-stimulatory molecules to activate T cells as well. In terms of history, so aluminum adjuvants, they were used for more than 80 years. They have a very long history, or 100 years, almost. And they are safe, and there are various different formulations to it. So most vaccines we have today, they are adjuvanted with different formulations of aluminum salts. But then in the 90s, some more other have been developed. So some of these ones are currently being used. So this is aluminum, but also other molecules to it. Here is nucleic acid, which can be used as well, and then also lipid nanoparticles. And here, for instance, aluminum, when it was introduced, Freon's adjuvant is very, very potent, but it's not used in humans, it's used in animal vaccines. LPS, very potent as well, not used as this form in humans, but a modified form in such as here, where is it? Here, a modified form of LPS, so AS04 is a detoxified LPS plus aluminum together. So as you can see, the development of those new adjuvants that are currently used, they are quite recent, relatively speaking. So in the 90s from now to now. So aluminum adjuvants, they bind close to antigens, and if you look into each different adjuvant, they will trigger different PRRs. So some will engage with TLR4, some will activate different types of C-type lectins, etc. But I guess the point for you here is not to memorize those, so I haven't included, but just remember that they induce those danger signals, and that's what would trigger the activation of the APCs. Here, just for your curiosity, there are quite a few formulations, so phosphate, hydroxide, potassium sulfate, quite a few different combinations, and they may be associated with other things. So the monophosphoryolipid A, which is the LPS, but detoxified. They are used in quite a few vaccines, as I mentioned, and it's still the most common adjuvant in the vaccines we use. With the delivery systems, and that's a bit heavy on reading here, but it explains well how they not only deliver safely the molecule, but also they stimulate the immune system as well. So they can allow sustained release. So some of these, they are, if you think about a stitch, right? When you stitch a patient, some stitches, they can degrade over time. They're absorbed. So here, some of these delivery systems, they are absorbed at different, it takes time for them to be absorbed as well. So they can actually deliver antigens in a sustainable release. So there are some experiments that are looking into that in stimulating the patient for a longer time. They can promote efficient uptake, so they can deliver directly to APCs, but because they are quite small as well, they can go to draining lymph nodes, and they can, which might be an issue in some instances, because one thing that we looked in COVID vaccines where mRNA was being going to. So if you look at, there are some discussions and some non-informed discussions like, oh my God, it's going to go into the ovaries and you're going to be sterile, you know, so, which is not true, but where those delivery systems go can be good, but sometimes maybe they shouldn't be there. So there is more investigation on those where they go as well. But if you look at obviously lymph node trafficking, that's a good thing. They can go via passive diffusion because they can stimulate cells there. They can do cross-presentation as well in dendritic cells, so that uptake, which most antigens could do, so uptake by MHC class 2 and then cross-presenting in MHC class 1 molecules and therefore activating CD8 T cells. And here are what we have currently used, which are the LPNs. So there is lots of data looking into the safety of those things as well. So looking again, increased bioavailability, targeting APCs, lymph node trafficking, and then cross-presentation in dendritic cells. Now this is just my last slide, so you bear in mind that in addition to the antigen and the adjuvant, there are many other components that might be present in a vaccine: preservatives, emulsifiers, stabilizers, and lots of trace components. So egg, it depends on where that cell or the vaccine was prepared. So if you think about, for instance, the rotavirus, you grow viruses in medium, and actually in the formulation you give to the baby is the medium that we have in the lab. You know, this example RPMI, for example, Dubelco media. So there are traces of things that, which is mostly fine when it's orally, but when it's injected, parenteral injections, then you have to think a bit more careful. Or if there are allergies as well to eggs or particular antibiotics. So here just looking at, no, this is the same image. It should be the different components. I'm gonna fix this image because it should be a different one here. But again, just think about those different components, and I think that's the last slide. So it wasn't quick, but at least I finished on time. Do you guys have any question? Yes.
Is monoclonal antibodies for spike protein in COVID can be regarded as a passive immunization?
Yeah, in the treatment, yes, because you can use MABs there. It could be regarded as a passive, but usually antibodies used in those other diseases that I gave as example, they will resolve the disease. For instance, in rabies, they will prevent rabies from progressing, right? In tetanus, they will prevent tetanus from progressing. Now, in COVID, using monoclonal antibodies won't necessarily prevent the infection from progressing, won't necessarily neutralize all viruses. You might have later complications, and they are much newer drugs. So they are not listed as a passive immunization, but they follow the same principle. Do you get what I'm saying? So they follow this principle of passive immunization because they're trying to neutralize the viruses, but they are not, I wouldn't classify them as under a passive, under the same class as the one that I presented, which we know that will definitely prevent the progression of those diseases. In fact, monoclonal antibodies have been used in various different diseases, not just COVID. In cancer as well is another approach, but they are not classified as passive immunization. Not at least not yet. Maybe one will come along and will be super effective, and across different strains of viruses, for example, and that could be classified as a passive immunization. But it's the same principle. Yeah. Good question.